Moving on to the morphology and of bacteria. Bacteria are classified into three types. Saprophytes. Saprophytes are the organisms which thrive on the dead and decaying matter. Saprophytes are the organisms which thrive on the dead and decaying matters. Saprophytes are the organisms which thrive on the dead and decaying matter. So they act as a natural scavengers on this earth. They act as a natural scavengers on this earth. They act as a natural scavengers on this earth. Saprophytes, they thrive on the dead and decaying matters. Saprophytes are the organisms. Usually they grow less than, and they are also called as psychrophils. They are also called as psychrophils. Mesophils, mesophils or human pathogen. They grow between 20 to 40 degrees centigrade. Thermophils, thermophils organisms which grow between 40 and 90 degrees centigrade. Psychrophils, mesophils and thermophils. Okay. Now important come. The organism which cause infection in man are called as pathogens. Pathogens. The organism which cause infection in man are called pathogens. The organism which cause infection in man are called pathogens. What is infection? Infection is defined as successful invasion and colonization of organism in human body. Successful invasion and colonization of microorganism in human body. Successful invasion and colonization of microorganism in human body. So when we get infected, it is suffixed with itis. It is suffixed with itis. So, what's your name? Vinay. If nose get infected, what we call it? Rhinitis. Eye become infected, we call it as ophthalmitis. Ophthalmology. Ear become infected, then we call it as ear. Call it as otitis. Otitis externa, otitis media and otitis interna. Brain get infected. Don't say brainitis, something else. What is it? Encephalitis. Encephalitis. Hair become infected. What do you call it as? Hair. I don't have, but we have plenty. It is called hair. Folliculitis. Bone become infected. It's called as osteomyelitis. If peritoneum get infected, it is called as peritonitis. Ovaries become infected, it is called as ovaries. It is called Oophritis. Testes become infected, it is called as Orchitis. If meninges become infected, then it is called as meningitis. If fallopian tube get infected, it is called as Salphingitis. If uterus get infected, it is called as, so endometrium is there, it is called endometrium. Ureter get infected, it is called urethritis. Urethra become infected, urethritis. Vaginitis. So you have to suffix with that itis, okay. So the quantitation of pathogenicity is coined by two terms as virulence. Ability of organisms to produce disease. Highly virulent, less virulent, moderately virulent. It is the quantitation of organism which will be able to provoke infection. Ability of organism to produce disease. Decreasing the virulence is called attenuation. Decreasing the virulence is called attenuation. Decreasing the virulence is called as attenuation. Decreasing the virulence is called as attenuation. Now, pathogens are the organism which produce infection. Clear? Pathogens are the organism which produce infection. So, infected organ is suffixed with itis. Infected organism is suffixed with keratitis is cornea, retinitis is retina, okay. Tongue, glossitis. So every organ is suffixed with itis, okay. Now there are something organisms which are called commensals. Commensalism. They live a symbiotic life in the human body. In fact, we have got more number of microorganisms than actual number of cells in our body. Human being is a walking, talking museum of microorganisms. Walking, talking, we have got trillions and trillions of microorganisms. Every part of our body is covered with microorganisms. Every inch of our body is covered with microorganisms. There are two areas which are sterile. Which are the two areas which are sterile? Like knowledge, brain. Brain and blood. There are two areas which are sterile. The brain and blood. Okay. Commensals, intestine is full of organism called Escherichia coli. Organism called Escherichia, everybody say Escherichia coli. Escherichia coli. Escherichia coli. Not Aishwarya Rai, Escherichia coli. Escherichia coli. But it is in an example, the advantages of commensals are 3. Hold it like this, 3. 1, 2, 3. Can you able to count? 3. Number 1, they produce intrinsic antibodies. Number 2, they produce intrinsic vitamins. And third is the most important. They are the competitive inhibition. They don't allow the pathogens to colonize and cause infection. They don't allow pathogens to enter, colonize and cause infection. It is called as competitive inhibition. The moment all the commensals are, and they help in the absorption of the nutrients. So usually the person who has got good number of commensals is usually quite nutritious, quite fat, nutritious. It helps in the absorption of the nutrients. But Escherichia coli is an example of opportunistic pathogens. Escherichia coli is an example of opportunistic pathogens. So 80% of the urinary tract, UTI, not Unit Trust of India, UTI stands for urinary tract infection. 80% of urinary tract infection caused by Escherichia coli. 80% of the urinary tract infection is caused by Escherichia coli and is common in females compared to males. It is common in female compared to males. UTI is common in females compared to male. You ask me why? Everybody? You ask me why? Why, sir? Yeah, good. Because short stature of urethra and proximity of urethra to the anal region. Short stature of urethra and proximity of anal region to urethra. And so depending on the temperature, psychrophil, mesophil and thermophil. Psychrophils less than 20, they are not naturally, they thrive on dead and decaying matters. Mesophils 20 to 40, all pathogens belong to mesophils. Thermophil, which survive high temperature. Psychrophil, naturally they are thrive on the dead and decaying matters. Mesophils are the pathogens. Pathogens are the organism which cause infection in man. The quantitation of pathogenicity is done by two terminology. Virulence. Highly pathogenic is highly virulent. When you decrease the pathogen, which we use in vaccination, it's called as heat killed, attenuation. Then commensals. Advantages of having commensals is intrinsic vitamins, intrinsic antibodies, competitive inhibition. They do not allow the pathogens to colonize and cause infection in human being. Next is they help in the absorption of nutrition. With this background, we will move on to the morphology of bacteria. It is made up of cell wall. Cell wall is made up of nam and nag. Cell wall is made up of NAM and NAG. NAM stands for N-acetyl muramic acid. N-acetyl muramic acid. N-acetyl muramic acid. N-acetyl muramic acid. And NAG stands for N-acetyl glucosamine. N-acetyl glucosamine. N-acetyl held together by peptide chain. Held together by dipeptide chain. What is the importance of cell wall? Importance of cell wall, it gives shape, it gives protection against the hostile environment, and it gives rigidity, density, protection, shape. It gives rigidity, density, protection and shape. Everybody. It gives rigidity, density, protection and helps to protect the bacteria. Cell wall is considered as tire. For the inside is made up of tube, which is a cytoplasmic membrane. It's like a tire. It's like a tire. So depending on the shape, bacteria are classified into cocci. Cocci are cocci. Coccus is singular, cocci is plural. Coccus is singular, cocci are plural. Hold like this, everybody. How many are this? 1, 2, 3, 4, 5. There are 5 important cocci which cause infection in man. How many? 5. The first one is staphylococci. The first one is staphylococci. Staphylococci is gram-positive cocci arranged in clusters. Staphylococci is gram-positive cocci arranged in clusters. It looks like bunch of grapes. It looks like bunch of grapes. It looks like bunch of grapes. It looks like grapes, not bunch, grapes. So you ask me why? Ask me why?
Why, sir?
Because they divide in all planes. Hold like this. They divide in all planes.
Why not?
And daughter cells fail to separate out from mother cells. Everybody. They divide in all planes. Not aeroplane. All planes. See, all sort of planes. They divide in all planes. And daughter cells fail to separate out from the mother cells. That's why they are arranged in clusters. That's why they are arranged in clusters. Next one is streptococci. Streptococcus means serpent. Streptococcus means serpent. They divide in single pane. They divide in single plane. They divide in single plane and daughter cells fail to separate out from the mother cell. They divide in single plane. Daughter cells fail to separate out from mother cell. They divide in single plane. Streptococcus means serpent. Streptococcus means serpent. Streptococcus means serpent. They divide in single plane. Daughter cells fail to separate out from the mother cell. Next cocci is pneumococci. Hold like this. It should be triangular, okay? Triangular. Next is pneumococci. It is called pneumococci because it infects the lungs, pneumonitis. It is called pneumonia. Pneumococci is lancet shaped or flame shaped. Lancet shaped or flame shaped, hold like this. Lancet shaped or flame shaped, two cocci enclosed in a single capsule. Two cocci enclosed in a single capsule. Two cocci enclosed in a single capsule. It is called pneumococci because it causes infection of the lung. Pneumonia, pneumonitis, pneumonitis. Infection of the lung, it's called as pneumonitis. Next two cocci are gram negative, they look pink in color. We'll tell you about gram staining. What is gram staining? One is half moon shaped. Half moon shape, hold like this, half moon. And two cocci, the plane end facing each other, plane end facing each other. That is meningococci. Meningococci. They are called meningococci because they infect brain. They are called meningococci because they infect brain. They are called meningococci because they infect brain. Next one is gonococci. They are kidney shaped or bean shaped. Hold like this. They are kidney shaped or bean shaped and concave end facing each other. They are diplococci. Concave end facing each other. They are diplococci. Concave end facing each other. So now start with me in a rhyme. Staphylococci. Streptococci. Pneumococci or gram positive, gonococci. What happened? You were shy or something like that? Gonococci, meningococci or gram negative, staphylococci, streptococci, pneumococci or gram positive, gonococci, meningococci or gram negative, staphylococci, streptococci, pneumococci or gram positive, gonococci, meningococci or gram negative, staphylococci. Staphylococci. Don't go further. Staphylococci. Divides in all plane. Looks like grapes. That's why it is called as staphylococci. Streptococci. Divides in single plane. It looks like serpent. It is called streptococci. Pneumococci is a diplococci, lancet shaped or flame shaped, broader end facing each other. Two cocci enclosed in a single capsule. Two cocci enclosed in a single capsule. Gonococci, kidney shaped and concave end facing each other. Meningococci, half moon shaped, plane surface facing each other. So now you know Staphylococci, Streptococci, Pneumococci are gram positive. Meningococci and gonococci are gram negative. Now we are stressing so much on gram positive and gram negative. What is gram positive and what is gram negative? Gram positive cocci are violet in color. What color?
Violet in color.
Gram positive are violet. Violet. What is that?
Violet.
Violet. Gram positive are 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 gram negative are pink. Gram negative are pink. Gram negative are pink. Gram negative are gram negative are remember your 2000 rupees note. They are pink. So why some organism retain violet color? Why some organism give away the primary color and take up the counter stain and look pink in color? This is explained by Hans Christian Gram. He discovered it is still considered as golden stain. I mean fundamental basics of microbiology. He devised staining and it is considered one of the most important staining technique. Whatever you do, whatever specialization you pursue, it's a lot of importance for identifying the organism, putting up the culture and putting up the antibiotic agents. Antibiotic and chemotherapeutic agents, antibiogram. So it's very, very important. So now, for example, what is the difference between 5 minutes more? What is the difference between gram positive and gram negative? Why some organism look violet in color? Why organism look pink in color? What is the difference between this? What is the difference between gram positive and gram negative? Now, don't be under the impression that cell wall gets stained. Cell wall do not get stained. The cytoplasm inside will get stained. Please remember. Cytoplasm gets stained, not the cell wall. Cytoplasm inside gets stained, not the cell wall. Now, the cytoplasm, there are 3 postulates. How many? Can you count? 1, 2, 3. 3 postulates. What are the 3 postulates? Number 1, gram positive cell wall is 40 times thicker. How many times? 40. 40 times thicker than the gram negative cell wall. Gram positive cell wall is 40 times thicker than the gram negative cell wall. 40 times thicker than the gram negative cell wall. But gram negative cell wall is more complex. It has got one more lipoidal layer outside the lipoidal layer outside the cell wall. Lipoidal layer outside the cell wall. We will continue in the next class tomorrow. Thank you.